All right, topic 10.2, video 2. Let's get to it. The polygenic means many genes. Right there. Human height is a classic one. Lots of different genes affect how tall you're going to get. And it's one of those that are affected by environmental factors, mainly nutrition. There has been a gradual increase in average height, in the past 100 years, largely because people tend to get enough food on the last 100, 120 years, whereas before, a substantial number of people just did not get enough. So here's a couple, a graph on human height. The average, you may be interested to know, the average female height is 164.7 centimeters. That works out to 5 feet, 5 inches. Now this is the median height, not the average height. Remember, those are different. So if you line up all women on Earth, shortest to tallest, the very one in the middle would be about 5'5". I believe the average is slightly below that. And then for males, it's 178 centimeters, or 78.4 centimeters. And that works out to about 5 feet, 10 inches. Again, I think that's about an inch taller than the average. But just polygenic traits, environmental factors. And here's a cool example. I love cats. Some of you might like cats too. But some traits like the Siamese cats can be controlled by how they interact with the environment, with the DNA in the cell, limiting available resources, so they can influence the DNA or influence the resources the cell needs to create that pigment or whatever that is. And think about what part of this cat, this cute Siamese cat, are that black color, that dark pigment. And if you think about it, it's the cooler areas. So on a cat, you're going to be losing heat from the ears, the front of the face, the paws, the tail. So these colder temperatures activate the gene to make that pigment darker. So only the darker areas express that fur color. Now, we had just in the last section, we did this Punnett square dihybrid cross. Remember, dihybrid means you're taking two parents. Sorry, messed that up. Two parents that have one of each gene. That's all dihybrid means. So they're both hybrids, basically. And you gotta be able to know what those phenotypic ratios are. So remember, 9:3:3:1. That's what that ratio. Now, that's the phenotypic. The genotypic ratio is different. That's all the different genotypes. So I believe there's 9 different genotypes. I'm gonna start writing them out. Big R, big Y, little y, and then big R, big Y, little y. Big R, little y, big Y. That's a new one. Big R, big Y, little y. 2 big R's, so we already have that one. Big R, little y. Do we have that? We don't. So big R, little y. That's this one. Here's the hybrid. We have that already. Big R, little y. We don't have that one, so I'm gonna add that. Big R, little y. Oh shit. Yep, we don't have that. Big R, big Y. We have that. We have this one. Little r, big Y. That's the new one. So little r, big Y. There we go. Little r, big Y, little y. We don't have that. And then we have this one. Big R, little y, little y. We have that. Little r, we got that. Little r, big Y, little y. So 1, 2, 3, 4, 5, 6, 7, 8, 9. There we go. So how many of this big R? I think there's one of those. There's one of those. Big R, big R. There's one. Is there another one? Yes, I believe there's two. Big R, little r, big R, little r. So there's this one. And there's that one. That's two. There's four of these. Got the big R. Let's see. Big R and then the little Y's. Big R's and the little Y's. I think it's just the one. Big R, little r, and then the little Y's. There's this one. There's that one. There's two. Little r, there's one. One. There's only one of those. How many of these? Oh, there's two there. So how much we got? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Thank gosh. So that would be your ratio: 1 to 2 to 2 to 4 to 1 to 2 to 1 to 2 to 1. Adding up to 16 total. All right. So that's how you would calculate that. I doubt on the exam I haven't seen anybody get that far. Another example, we see, this dihybrid cross here. We're looking at purple flowers. See, one of each. Big T, big P, little p. Okay, both tall and purple. And then if we do the Punnett square, oh, like a magic. Here's the Punnett square, showing you the phenotype, the 9331. When it says refer to the Punnett grid, basically, do what I did here, is you just count up all the different ones, and there's 9 different types. So 9 types, 9 varieties. Okay, you can go check that out. So being able to identify the recombinants. Here are the parents here, like the P1 generation. Parents, you don't see any allele that has that's heterozygous, right? They're all homozygous. They're both, like these are both. Let me write that. They're both little B's and A's or capital B's and A's. Now if you look down here, this one is a combination. This is a combination. You can see these B's switched. This is a recombination. And over here, let me look. Yeah, these are the same as the parents, so they're not recombinants. So the same. These are not recombinants, because they're the same. Now, this idea of recombination really took hold further, in Morgan's fruit fly experiment. So fruit flies are used a lot to show linked genes, recombinants. Here what we have is we have a heterozygous individual. You see B plus B, VG plus VG. So heterozygous for both traits. In both traits. And here we have a homozygous recessive. Got all the recessive alleles for body color, and wings. And then if you normally put them into a dye hybrid or a Punnett square, looking at two different genes, 4x4 Punnett square. If you did that, you would expect an even number of all of these. That would be your expected phenotypes, basically 1 to 1 to 1. But when they do this, when Morgan did this, look at what the actual observed was. It was way higher for those parental phenotypes. So when you see that, when they look like the parents, when the babies look like the parents, when they all shouldn't, you know you're looking at linked genes. Those genes are linked. The two genes for body color and for the wings. Here's another little practice with recombinants here. If we recombine to kind of switch things around, we get this instead of, look, all these are homozygous, right? Either dominant or recessive. But to recombine them, to have crossing over where this one goes over there, this one goes over here, we'll get something that looks more like this. Oops, sorry, it should be a lowercase t. My apologies. So that's just, you just gotta, on the test, they're gonna show something that looks like this. A little line with letters on it, that's an allele. And those are the two genes. So this is the allele. And the genes on allele. Okay, on that. Actually, it's a chromosome. Sorry, the little B's are the alleles. I'm sorry. That's the little piece of the chromosome. And those are the alleles. The versions that are on that gene. So here we got, here we have a little cross between these gray normal, like you saw before. These would be considered the normal offspring, the ones you would have. And if we had any of these recombined, you'll see, you don't see this in any, this doesn't exist up here. It doesn't exist up here either. Does not exist. Yet that is, because these did switch somehow during meiosis in that crossing over event. And these would be the recombinant offspring. Because they have alleles that have recombined during that process of meiosis. So chi-squared test, I think I mentioned it before. Yeah, it's confusing. So painstaking to do it all. You probably, I doubt it you'll ever do that on the test, but you'll have to analyze it. So let me just show you. Basically, you're comparing your observed data with what you expect, and this is a way to see if these genes are linked. So remember, in a normal dihybrid cross, we should be getting that 9:3:3:1 ratio. Okay, but in the experiment, this is what we actually got. So we need to see, does this fit? It looks like it might, right? This is the highest. These are pretty similar. This is the least. So it might fit expectations. Sorry about that handwriting. It might fit expected ratios, but we need to see if it's close enough. we use Chi-square to test that. We did this last year, M&M lab. I think I mentioned that before. We did a little sheet, filled it out. So remember, null hypothesis, alternative hypothesis. Make sure to always, we're going to say there, oh, some nice birds out there. Oh, that's a nice little yellow finch. Oh, I missed that. In quarantine, I'm checking these birds out all day. Anyway, no hypothesis. There's no significant difference between observed and what we expect, so they're not linked. But if it is different, if our chi-square value is above our critical value, then we know it's linked. So you first start, you make a table, and we have our observed here, and this is what we got from our experiment. Our experimental data. And this is what we would expect. So what we do is take the total, you can see here, multiply it by the ratio. 12/16 times 9/16 should be getting 684. 228. Because we take, remember, that total of all the different individuals we had in our test times it by that ratio. So now that we have that observed and expected, we want to put it, eventually we're going to use this calculation where we take the observed minus the expected, square that number, divide it by the expected, and then we're going to add that up. It'd be 1, 2, 3, it'd be 4 times for each of these. 1, 2, 3, 4. So observed, expected, observed, so 701 minus 684 gives us 17. 204 minus 220 gives negative 24. And you can see how it goes. I said this is not hard, but it's confusing. 243 minus 220 is 15. 68 minus 76 is negative 8. So now what we do, we square it, which gets rid of the negative, and then we divide it by expected. So we're going to square this, divide it by this number, and we do that, we get these 4 numbers, and all we got to do is add those up. So this plus this plus this, put the equal sign, grab my handy. I gotta tell you, my TI-30XS multi-view calculator, very nice. The buttons are so good, positive feel on them. I really hate using, I don't hate, but that's a strong word, using iPhones for a calculator. It's a real hassle. So I got all of them added up. I add them all. My critical value, or my chi-square value, is 4.78. So chi-square is 4.78. Ta-da! But you're not done. 4.78. You got to figure out your degrees of freedom, which is basically how many outcomes do you have? How many possibilities? There's 4. You take that minus 1. So your degrees of freedom is 3. And we look in a table. This has, it goes 1 through, I think, 20 degrees of freedom. And what we want to do in science, we're really looking at this 0.05 level. So if our number is bigger than this, then we're going to reject our null. But we see our number is not bigger. So chi-square value is less than the critical value. And I should have put that. So chi-square value of 4.7 is less than the critical value of 7.815. So we accept our null hypothesis, which is the genes are not linked. They have to be different enough from that expected. And remember, what was I going to say? Okay, I paused it to try to remember. I still can't remember what I was going to say to you. But, yeah. Oh, that's what I remember. Oh, good. The numbers are never going to be exactly expected. In fact, if numbers are ever exactly what you expect, be very, very suspicious. So they're never going to be 9, 3, 3, 1, just random chance. So we need a test like this to figure out how far away from that expected value do we have to go to have it mean that it really isn't what we expected, and therefore probably linked genes.